So
That's funny. That's funny.
they each have their own color. Like one boy's all his stuff is red, one is all
That's so
blue.
funny. So did we. So my brother's name is Jared, so he wore red, Jared, and then I'm Dean, so I wore green, and then the other one just wore whatever he wanted. So
That's hilarious.
I love that.
Awesome. Well, I think we'll get started. Some people may still be trickling in. But hi, everyone. For those of you who don't know me, my name is Samantha Atlas. I'm the research program coordinator here at the ADRC. I am very excited for our April talk featuring Dr. Logan Dumitrescu. Just a little summary. Dr. Dumitrescu is a computational human geneticist and assistant professor of neurology at Vanderbilt University Medical Center and a core faculty member within their Memory and Alzheimer's Center. She leads the computational neurogenetics team there, as well as the research education component at their ADRC. Formerly trained in human genetics and genomics, Dr. Dumitrescu's research goal is to investigate how these genetic variations can impact different phenotypes, specifically looking at Alzheimer's disease and looking at and characterizing sex-specific genetic drivers of Alzheimer's risk and resilience. So you guys have been here before, but if you haven't, how we'll do this is we'll do the talk and then we'll save about 15 minutes afterwards for Q&A. Feel free to raise your hand, put them in the chat, unmute, anything like that. But we'll get started.
Great. Thanks so much. I'm really excited to be here. I recognize a few names. Hi, Rick. Hi, Lindsay. So hopefully you guys call me out if I say anything incorrect, please. And let me start sharing. All right. We're good. Thanks, Dean. Appreciate it. All right. All right, yes, as Sammy mentioned, I'm here to talk about sex-specific genetics of AD, and I'm over here at Vanderbilt where we're having a balmy 85-degree day, unlike in Boston, which I'm slightly jealous of. So hopefully let's see, make sure I can transition to the next slide. Yes. All right, so this is a very brief outline. I actually had a really difficult time putting today's talk together because there were so many things I wanted to discuss, actually. So if I end up going a little fast or talking too much, feel free to interject with questions or any of that. Sometimes my enthusiasm can get a little overwhelming. But I'm going to start talking about sex differences in AD. I think you guys already got a preview of this, so I won't spend a ton of time on it. Dr. Amelia Peterson from our group, I believe, spoke in December on her work. So I won't spend too much time. Then I will get into some fun genetics and the genetic architecture of AD. Then I will talk about the reason why we like to study endophenotypes of Alzheimer's disease. And I will do a huge plug for our Phenotype Harmonization Consortium, which I'll talk a little bit more about. And then lastly, I'll just give you some examples of some of the work we've been doing on finding these genetic and genomic drivers that are sex-specific. All right. So while there are well-established differences in sex differences in the prevalence and the incidence of many neurological diseases, and I really like this figure, you can kind of see there's this whole spread, and it's not just male-specific or female-specific. You can see there's a big gradation across many different traits and disorders. But of course, we have Alzheimer's disease way over here on the left. And that is because there is striking sex differences in Alzheimer's disease, where we have females make up 2/3 of all of prevalent Alzheimer's disease cases. And I think Amalia probably showed this slide already, but just to remind you that there are differences in lifetime risk of Alzheimer's disease. So let's say we take somebody at age 45. If you're a woman, you have 1 out of 5 chance of developing Alzheimer's disease. Whereas if you're a male, then you have a 1 out of 10 chance of developing Alzheimer's disease. And these risks also go up by age 65 as well. But the sex differences still remain. So both men and women can develop Alzheimer's disease, right? But women seem to be more vulnerable. We see a lot more women with AD, but are they actually more vulnerable? And this has actually been highly debated. They've had I-START debates from the PIAs, the sex and gender PIA. And I don't have time to go into it, but I just wanted to give you a little taste of kind of the debate that's going on here. So why would women be at higher risk? Okay. for one, women live longer than men, right? So here we have a graph. This is life expectancy. This is within the United States. And how life expectancy for both males and females has changed over the last few decades. And this graph shows you two things. One, men and women.
You gonna do a sandwich, baby?
Oh,
You gonna
oh, great.
Sorry, just was me.
No worries. Okay, so two things. For one, the graph is showing you that, yay, we're living longer. Both men and women are living longer since 1790. So that's a great thing. But then you'll also see that there's this widening gap between men and women over time, with females consistently living longer than males. Okay. Now, this is not unique to the United States, and many of you may have seen this this graph too before, but women across the world in pretty much every culture with every different kind of economic, social, environmental breakdowns, women live longer than men. So you'll see there's this blue line, and anytime any of these countries, which I think are size is probably what the dots are, if they lie above the blue line, that means that females are living longer. And you can see there is no dots below the blue line, which would mean that the males in that country are living longer. So women live longer than men. And what is the greatest risk factor for Alzheimer's disease? Age. But of course, it's not that simple. And the differences in risk of AD between men and women is not consistent around the world, actually. So for this, if you look at the top graph here, this is a pooled data from European countries, and you can clearly see there's this divergence between male and female number of cases of AD dementia, probably about age 78, but definitely past age 80, you can see there's far higher, newer cases of Alzheimer's disease in women than there are in men. But then when you drop down to this other data within the United States, I believe this is from the Mayo Clinic in Rochester, we don't really see much of a difference, and not that huge split like you're seeing in some other countries. Another reason this is not so simple is, and something we deal with or haven't quite exactly figured out how to deal with, is survival bias. Older men actually may be healthier and at lower risk for AD because they have survived other risk factors and have lived beyond that. So an example of this would be cardiovascular risk. Males are at higher risk for dying from cardiovascular disease compared to women of the same age. So men that live beyond 65 have made it maybe past that higher risk from death from cardiovascular disease, or will actually be healthier than the women of the same age. So I didn't even get a chance to go into the nitty-gritty, but you can kind of see there's some discrepancies here and some confusion, some mixed findings in the literature. So really, it's are women really at risk? There's also a lot of debate whether women are more susceptible versus more resilient. And so, given all these uncertainties, should we actually still look at these sex differences, or do they matter? And of course, I would argue absolutely. And the reason for this is that men and women develop the way and the why they develop AD may be different. And so why they develop it, and then there's evidence too that their response to amyloid and tau, to neuropathology, the response to these neuropathologies in the brain is also different. I'll give a couple examples of that. So females have more AD pathology at autopsy, and this is seen over and over and over again. This is even after controlling for age and education levels and some other factors. Women consistently have more AD pathology at autopsy. Women also decline more rapidly on a cognitive scale. So this is some global cognition looking at males and females, and they decline more rapidly. You'll see they start out higher, but then once that transition kind of is made, they rapidly decline. And again, so this goes back to are women more susceptible, or are they actually showing resilience, right? So can women tolerate this pathology longer than males before there's essentially a switch is flipped, right? And they just cannot overcome the amount of burden. Another important fact is that women with Alzheimer's disease live longer than men with Alzheimer's disease. Okay, so there's probably many reasons why there's all these differences, and we really honestly don't know exactly what's driving it, but because there's many factors, right? So we've got socioeconomic factors. I would call these gender differences. So differences, of course, in education levels, occupations and behaviors, environment and cultural social influences. But of course, as we all know, there's actually biological differences too, right? We've got hormonal differences, and women undergo phases of life that men do not, including pregnancy and menopause. And then, of course, there's my favorite, which is genetics. And the most striking is indeed that women and men have different sex chromosomes, with women having 2 X chromosomes and males having XY chromosomes. So we were talking about risk factors, right? And the Lancet Commission puts out this really nice paper every couple of years talking about modifiable risk factors. And I think in the 2024, they estimated that about 45% of all dementia cases worldwide could actually be prevented or delayed by addressing, and here they have 14 of these modifiable risk factors. So, I mean, that's pretty amazing, right? But of course, there's something missing there. What about the non-modifiable risk factors? And that's what we're interested in. So I already mentioned age, greatest risk factor for Alzheimer's disease. I've mentioned biological sex. Women are at higher risk generally, but also genetics. So now we're going to talk some genetics. All right, so Alzheimer's disease is highly heritable. So the heritability is really just the proportion of phenotypic variance or risk for a disease that's due to genetic factors. If you'll see here, there's this nice pie chart where here they estimate that AD risk is essentially driven by either modifiable or environment risk factors 35% of the time. And then you have a range of heritability from about 60 to 80% of risk factors are due to genetics. Now, to put that in comparison, let's say, so height is highly heritable, right? We're talking 80 to 90% heritability. But then when you look at other common complex diseases, the heritability is usually not this high. I believe with cardiovascular risk, we're going somewhere from between 30 and 60. So the fact that Alzheimer's disease has a really high heritability makes it a really great candidate for studying the genetics. I hope I can convince you of that. So let's delve into that little green piece of the pie here. What exactly is contributing to this heritability? Well, so we have, let's see if I don't know if y'all can see my, can y'all see my mouse or do I need to?
We can see the cursor.
Great. So, so this 1% here, these are the familial risk disease. These are this is APP, presenilin 1 and presenilin 2. So these cause the familial form of Alzheimer's disease. This is the early onset aspect. This really only accounts for less than 1% of AD cases. So then next you'll see another piece of this pie is apolipoprotein E or ApoE4. And the E4 allele actually contributes a pretty large risk here. And I'll go into this a little bit more later. Here they estimate about 5 to 9%. But pretty much the majority of the rest of this pie, we're talking about known AD loci, other SNPs in general, and then other genetic variation. And this could be a myriad of things. Outside of single nucleotide polymorphisms. But I want to delve into this aspect here about looking at SNPs and GWAS to find genetic drivers. So I don't know I hope I'm not talking down, but we're going to talk about genome-wide association studies. And this is really a workhorse for human geneticists. This is how we study common complex diseases, right? So we're looking at millions of variants and their association with a trait. Here I have a Manhattan plot. This is from the Lambert et al. Paper, very definitive paper here. And you can see on the x-axis, we have chromosome position. So this is just how we rank the variants, basically just on position where they lie on the chromosome and in chromosomal order. Then you have on the y-axis, you have the negative log of the p-value. of the association with that trait or with that risk factor. And the higher the number here, the more significant we get, right? So for one, you can see we have a very strong signal here for ApoE, right? We have APO, we have this long line that's drawn from all the variants that are in high leakage disequilibrium on this locus of the ApoE locus. This is kind of our positive control in a lot of our studies as well. And then we have multiple other loci here, and I didn't even put some of the larger GWASs on here because this Manhattan plot starts to get so crowded. It's actually hard to see and delineate some of the genes. Oh, I forgot to mention, if you aren't aware, the reason we call it a Manhattan plot is because it's supposed to resemble the Manhattan skyline, right? So you want those tall skyscrapers, like ApoE here. So there's been a huge amount of research actually using genome-wide association studies, looking at AD risk. And we have gotten up to over 1 million participants where we've looked for genetic variants. And these are common genetic variants across the genome. And collectively, we've identified, I think the last estimate I could pull was about over 100, just a little bit over 100 independent variants over 81 loci that are associated with AD and dementia. This is, of course, only in European ancestry populations. So that seems like a lot. We know a lot, but there's lots of so GWAS is very much for discovery, right? So we're discovering new loci. And one of the reasons that it can be difficult is that a lot of the SNPs, the loci that we find, end up in areas where it's hard to tell what gene is possibly driving the association that you're seeing. For one, I have an example here up at the top. So both of these are locus zoom plots from that Manhattan plot that we saw earlier. So we're zooming in on a chromosomal region from that larger landscape, right? So here we're zooming in on a chromosome 19 loci. And you kind of see here, okay, we have our top SNP labeled here and different color with the LD. But look, down here, there's a gazillion genes, right? So where does this variant, what's driving this association? And we know now this is ABCA7 driving this. But you can see this is a very dense genetic region. You could see something a little bit different here when you look at the BIN1 locus on chromosome 2 when you zoom in, while the variant over here is the one that has our strongest signal, our strongest association. It's actually outside of BIN1. It's close, but it's not within a coding region, right? So a lot of these variants end up this way, gene dense regions, non-coding regions. And really, if we want to really be able to narrow these down, we need sample sizes and we need racial ethnic diversity. So we need genetic ancestry diversity for fine mapping, which we're working on, but has been very difficult. So basically our group has thought there's got to be other ways to get not only to find novel loci, but kind of hone in on what loci are driving some of these associations. And we do that with two different ways, or actually we combine kind of two different methods. And one is sex-aware analyses. So essentially, we hypothesize that sex differences that we see in Alzheimer's disease can be understood if you actually look for them. Surprisingly, this has gotten better, but surprisingly, a lot of genome-wide association studies were not looking at sex differences. They did not stratify by sex. They would often covary for sex, kind of regress that effect away, So we want to actually specifically look at sex when we can. And then I kind of already previewed this a little bit. We are going to use endophenotypes, right? So these endophenotypes could be genetically simpler, where they actually may have larger effect sizes and be more proximal to gene function than, say, an 80 case control outcome. And of course, 80 case control outcomes can often have misdiagnoses. They also, what about where does MCI fall in there? So there's a lot of reasons to look at sensitive continuous traits that actually explain some of these Alzheimer's disease risks. And one way we do this is through our phenotype harmonization consortium. Now, just a little bit of background here. There's been a huge explosion in data of Alzheimer's disease, right? And I'm talking huge amount of genomic data. This is genome-wide association studies. We have whole genome and whole exome sequencing as well. Just very large numbers. But then not only on top of that, these endophenotypes, the technology has advanced so much that now we have all these molecular biomarkers that we can use, both brain, PET and imaging and blood and cerebral spinal fluid. And we can combine these with some of our others more standard endophenotypes like MRI and cognitive outcomes to maybe hopefully drill down on what's going on in the etiology of the disease. These numbers were getting so big with all the phenotype data that the NIA actually put out a funding opportunity announcement. This was in 2020, I believe. And I just kind of wanted to highlight here what their reasoning was. But they essentially were asking for people to come in and put in a U24 application to harmonize phenotype data. So here they recognize that there's massive amounts of it, that there's a challenge to analyze across studies because data are similar, but they're not the same. And so the Phenotype Harmonization Consortium was born. And we have 4 aims for the PHC. It's really to curate the data. And this is a lot of work. This is getting all the data from different institutions, ADRCs, a lot of data use agreements. And then we have the harmonization step, which I'll talk a tiny bit more about. And we're also tasked with disseminating this work to other researchers inside and outside of the ADSP and to educate individuals, not only on how to do data harmonization, but like I'm doing right now, the importance of data harmonization. This takes a very large team. You can see here. So I work with Dr. Homan here. I was part of the coordinating center, but we have different domain leads for all of our different endophenotypes, whether it's diffusion MRI, structural, or PET. We've got neuropath, vascular, fluid biomarker. We coordinate with the Million Veterans Program and the Charge. And then you may notice somebody over here from your group, for Jesse Mez, who helps with our cognitive harmonization. So I'm going to talk about that too. And I didn't see Jesse on the call, but if he is, but Lindsay, please call me out if I say something wrong. So Wyatt is...
I'm here, but I have to jump off in 5 minutes.
Oh, hey, Jesse. Okay. This is going to be a very simplified version. So if you guys have questions or complaints, send them to Jesse. All right. So regardless of studies, most participants that are studying in an aging Alzheimer's disease study are going to undergo a clinical workup, right? And it's a battery of neuropsychological testing. They generally all use the NIA AA criteria for an AD diagnosis, but there can be at least big differences in the extent of that cognitive measurement, right? You can go from a 6-item screener to a multiple-hour cognitive battery. So there's a strategy that's needed here to not only be able to pull data together, but then also to facilitate replication, which is especially important in a genetic study where replication is kind of like the gold standard for an association. So let's go through a couple examples of some challenges. One is different test items, right? So the ADRCs use and collect a standard form. This is the uniform data set. Maybe many of you have heard of this or been on this. I think we're up to UDS4 now. But this was an interesting study by Crane and Jesse, where they were looking at comparing the UDS, the first and the second UDS, actually to the UDS3. And while the first 2 versions were pretty similar, the UDS actually introduced some non-proprietary cognitive tests. They changed things up, tests they wouldn't necessarily have to pay for. So like they substituted, for instance, the MMSE for MoCA. And they have there's a correlation over here that compares some of the different item types and how related or how good they are. And you can see the correlation is, it's about 70 to 80, which is good to very good. But So what does that matter? Well, it does in the end when we're putting together these cognitive composite scores like memory of or executive function or language. Version of the UDS actually matters. You can see here the two different versions. We have blue and then green. Even at all the different type of diagnoses here across the board or cognitive domain, you're going to see some differences depending solely based on the test that was used. And that's just one example of other challenges. There's a ton more. There's noticeable differences in cognitive performance based on education level, sex and gender. Women have higher verbal memory performance usually than men, for example. There's cultural differences. Try naming a camel if you've never seen a camel. Mode of administration. This especially came up during COVID, where a lot of the in-person testing had to be replaced with telephone cognition. So that how do we reconcile even the same person over the same study that if they've been interviewed in person over versus another time over the phone? And then of course language, which I know our cognitive harmonization group is going well into now that we're having and increasing the diversity of a lot of our cohorts. Okay, this is where I'm going to trip up, Jesse. So, so how do we harmonize? How do we harmonize these really complex data? And really, it's they take a modern psychometrics approach. So if I understand right, this is essentially a lot of the same kind of math and behind when they're standardizing, say, SAT scores, right? They're taken across the country, different scores, but they have to all in the end be on the same metric. So this is the same kind of math that they use there. But essentially what happens is we get a panel of experts together. They look at all the different, let's see, here we have an example of 4 different cohorts, ROSMAP, ACT, NAC, and ADNI. Look at their different items that they use to measure memory performance and put them in, once you put them in domains, so once you decide that this is a memory performance item, then you identify these anchors, and these are anchor items. These are tests that are either administered consistently, so it's the exact same test across, or the test gives you the same amount of information. And these are really the core and how they can facilitate this co-calibration. Then they do some math that I don't quite understand, which is some confirmatory factor analysis, and they look at single or bifactor models. But essentially what comes out of this is a really nice continuous metric of a cognitive composite that across all of the studies included, and they're all on the same scale. They're all on the same metric. So for the ADSP right now, we've harmonized, we're getting close. I think these numbers are a little outdated because I think we're closer to 500,000 visits, but we have 30 cohorts, more than 5,000 variables. This is contributed to manuscripts, more than 100 manuscripts and abstracts. We have a lot of data. We've got the endophenotypes we cover. There's 8 of them are related to cardiovascular risk, cognition, fluid biomarkers, and then neuropath and neuroimaging. And if you want to learn more how to, like what we're doing, and if you want access to the data, please feel free. That I have some QR codes here that I think still work. I checked them yesterday. All right, that was a lot. We're about half, and I'm a little more than halfway through here. So we're gonna switch it up and talk about some of our genetic findings, unless anybody has any burning questions. All right. So first, let's talk about that gene I mentioned earlier, apolipoprotein E. So ApoE is the strongest genetic risk factor for Alzheimer's disease, for late onset, I should clarify. So you'll see in this graph is actually on our x-axis, we have minor allele frequency. So how often do you see the risk allele in the population, right? So not very often, all the way down here to much more common, all the way on the right. And then we have effect size, essentially, on the y-axis to odds ratio to highly penetrant, which is essentially if you have the variant, you will likely develop the disease. And we see these are the three genes that I mentioned earlier that result in familial Alzheimer's disease. So APP, presenilin 1, and presenilin 2. And then you'll see, I just really love this graph, and ApoE is so unique to me in a lot of ways. When you're studying a common variant, often what we see when we're looking at GWAS results is variants that fall here. So you see these little blue dots? These are variants that have very small effects, right? So they have odds ratios of 1.1 or so, and very small effects, but they're very common, right? And in the population, right? ApoE is just a very unique variant here in that you can see it's common. It's maybe not as common, but it's common in the population. And its effect size is huge, which is very rare. All right, so ApoE actually shows sex-specific effects. And we've known about this for a good 20 some odd years, maybe longer, and that the E4 allele shows a stronger association, so higher risk in females than males with the same allele. And we've actually looked at the effect of ApoE with our endophenotype data, right? So that was case control. Now, what about some of the other endophenotypes that are connected with Alzheimer's disease? So first, we looked at cerebral spinal fluid, right? And looked for sex differences with this ApoE4 allele to see is there a difference between the sexes? Does ApoE have a stronger effect in males or females, or not at all for that specific neural path pathology, excuse me. So when we looked at that, we were actually quite surprised by this. We saw an effect, a sex-specific effect with ApoE4 and tau, and this was stronger in females. That wasn't the surprising part. For us, the surprising part is we did not see it with amyloid. We didn't see any sex-specific effects. The effects were essentially the same between males and females when we're looking at CSF amyloid. So Dr. Rachel Buckley has also worked a lot with ApoE and looking at sex differences. And she did a really neat project a couple of years ago where she looked at amyloid PET. So we had CSF, she's looking at amyloid PET. It was a small sample size at the time, but she did not see an association either. Funny enough, she also looked at cognition, and she did not see a significant effect until she actually took into account amyloid pathology. And she saw that there was, in fact, a sex-specific effect driven by females, but only in individuals that already had A beta. So they were A beta positive, which is something we actually noticed ourselves in our CSF measures. So we have, a couple findings here, right? So we're not seeing sex-specific effects of ApoE with amyloid. We are seeing it with tau, and there's evidence in cognition. And then we did an even larger study where we looked at those different cognitive composites and looked for sex-specific effects with ApoE. We replicated the memory finding, but we also extended this to language, right? So this those ApoE findings kind of started this theoretical framework that other people have actually noticed as well. And not only do we posit that genetic factors act in a specific manner and that they can drive the presentation and progression of AD, we hypothesize that a lot of those genetic factors are actually downstream of amyloid. So those would be more take place with tau, with cognition, and with actual disease risk, and are stronger in the presence of amyloid pathology. All right, so what if let's go beyond ApoE. We talked about GWAS. Let's look at some GWASs. Again, I've mentioned that these are for discovery-based, and we have several examples. So I'll give you my favorite one first. This was actually one of the first ones we did, and it kind of blew my mind. So you'll see the panel on the left. This was a genome-wide association study for CSF tau levels. And this is out of Carlos Cruchaga's lab. And you see our friend here, ApoE with the blue, but he also identified a couple of novel loci, including this one here on chromosome 3. You'll see this, again, this skyline, you've got this big tall building here, right, on loci hit on chromosome 3. So we actually took that same data and worked with Carlos and ran that GWAS sex stratified. And so this is a Miami plot, unlike a Manhattan plot. So we essentially, it's flipped, right? So you could almost imagine in Miami where there's a skyline, but then there's water, right? And so you see the reflection of the water. So here we're comparing our male and our female associations. And what we found was that chromosome 3, seen in him blue, was totally driven by females. It was only seen when we looked at the females specifically. There was no association in males with that loci. Now, this was really cool, and we can use this to kind of delve down a little bit on what's going on at this locus, right? So the previous paper had, indicated that the nearest gene was this GMNC, because the locus, falls right there. But this was surrounded by numerous genes, and the evidence for GMNC wasn't super strong. So what we wanted to do is we actually looked at gene expression in the dorsal lateral prefrontal cortex in ROS-map participants. And we looked at 18 genes in that general locus, about a megabase on either side, to see if we could actually determine what gene was driving this genetic association. And we noticed that brain levels of 2 different genes, not GM and C, so it's this Claudin-1 and the Austin gene, were associated with tau, and in females only. So this gives us a little bit more of a direction to go into. Maybe these are the actual driving, one of these are the actual driving causal genes. And we've done something similar for looking at neurofibrillary tangles at autopsy. Here we actually found a male-specific loci here. This one was really interesting because we were not able to pin down what gene was driving this, but another group came in and independently verified that the gene, the implicated gene here was this T-span13, which has actually been shown to be involved in processing of APP, right? Very cool. We've also done this for cognition. So this is a recent paper that actually just got accepted, where we looked at sex-stratified GWASs of 3 domains of cognition. So we had the memory, executive function, and language. And a very cool finding came out from our language performance GWAS. Here there was a SNP located, you can see here in the locusome, it was all female specific. And it was downstream of VRK2. And we had some EQTL evidence that was also driving that the gene here, the causal gene, could possibly be this VRK2. But what was even more fascinating and kind of, it's an abundance of evidence when you're trying to figure out a causal gene. One was this gene keeps showing up in a lot of other GWASs that have to do with neuropsychiatric traits or with musicality and beat language. So you'll see here we've got musical beat, stuttering, language connectivity, along with neuropsychiatric traits of bipolar disorder and schizophrenia. So we are fascinated by this candidate gene, and we're working with another group here at Vanderbilt to kind of maybe drill down to see what's going on with this gene. It is a vaccina-related kinase. It regulates a lot of things, but multiple signal transduction pathways, and sometimes these pathways can affect neuron growth and migration. So again, this is a fascinating candidate that we're kind of drilling down more on. And then lastly, I think I've got a little bit of time. I want to talk about our jump from DNA to RNA and looking at transcriptomics and sex differences and endophenotypes of Alzheimer's disease. Okay, so we know that actually sex plays a large role in gene expression, right? So there was a big study in 2020 by Olivia et al. Who sampled this using the GTEx data. So I think there was over 40 tissues here, and they looked at sex differences, right? And the sex biased genes that were overexpressed in one sex versus another. And they found that 37% of all genes are sex biased in at least one tissue. And I've highlighted here, they did see that with, I think there's 10 brain tissues that they looked at. You could see that there's definitely some sex biased genes, right? And there have been studies, sex-specific transcriptomic studies of Alzheimer's disease. But again, they're usually just comparing an AD brain at autopsy versus a non-AD brain at autopsy. But they haven't really associated that with endophenotype. So we wanted to dig a little bit deeper on some patterns we may be able to see with sex bias expression in and our AD traits that we're interested in. So here again, we're using our ROS-map data, about 767 participants. We've got about 1,800 genes, and ROS-map had sampled 3 different regions of the brain. We have the caudate nucleus, the dorsolateral prefrontal cortex, and the posterior cingulate. And our 3 outcomes of interest here were, again, we're going to have amyloid burden, tau burden, and we have measures of cognition across time. This is a big, busy table. What I really just want to focus on is that we did propensity score matching. And so we took a whole bunch of different factors, and we wanted to make sure that we had equals number of males and females, and that they matched on all of our important covariates, such as age at death and education, basically because we wanted to directly compare the number and the extent and the effect sizes of associations in males versus females. And so we didn't want sample size differences or covariate differences coming in there. We wanted to make sure that we started on equal footing there. So we just ran some, these were simple linear or mixed effects models where we had our outcome, so amyloid, tau, or cognition, and our gene expression. And we looked at within all our individuals, and then we would stratify by sex and looked for sex interactions as well. And we looked across all the autosomes. We have X-linked genes and Y-linked genes in there too, which I don't have much time to go into some of the amazing work that we've done, that other team has done, looking at the X chromosome specifically. So what did we find? Okay, so we have a circle plot here, and each line essentially represents a significant association between a gene and a trait, and it is broken down by the 3 traits on the top and by the different tissues on the bottom. And it's also broken down by whether there were main effects or sex-specific effects. So everything in that green tells you were the main effects. And you can see just, if you step back and take a big view of it, we have a lot more green than we have the male or the female pink and blues, right? So we have a lot of more main effects with our outcomes of interest than we did sex-specific effects. So really in total, it was about only 10% of these associations were sex-specific. But let's delve deeper into that 10%. So when we look at those associations, we see an interesting pattern here, which reflects kind of that theoretical framework I talked about earlier. We see a lot of associations, if you look up top here, with tangles and cognition. And we see very few associations with amyloid. So a lot of our sex-specific effects are downstream of amyloid, we would argue. You also see a lot more pink than you do blue. Right? So let's break this down. Of the 2,000 or so sex-specific associations, 73% were female-specific, hence all the pink. 90% were with tau and cognition. And 4%, only 4% were X-linked. But this is actually consistent with just the percentage of how many genes are on the X versus the autosomes, right? So we don't see any enrichment there. What we do see enrichment for is actually genes on the X that are known to escape X inactivation, which I'd be happy to talk to later, but didn't have time to get into here. And another thing that we saw is we didn't see a lot of the same genes across all 3 different tissues, which I was very surprised at. but looking back, it's probably not all that surprising. There was only 16 of these genes that were significant in 2 or more brain regions, and none actually, again, across all 3. And then another cool thing you can do with these transcriptomics is you can do these gene set enrichments, and you can look for pathways and whether genes are upregulated together, downregulated together. They can tell you some more about the biology. Here we found some upregulated genes that were in pathways related to neural development and immune-related pathways, not surprising. So essentially this can tell us, we're trying to drill down, not only we're getting a bigger picture of how much does sex affect gene expression and what pathways are involved as well. So very cool stuff, I think. And so I know I'm running out of time. So I'll do a quick summary. Hopefully I've convinced you that there's more women than men that have Alzheimer's disease, but there are differences in how men and women respond and probably how they obtain these differences in AD neuropathology. There's risk factors, both genetic, and we didn't have a chance to go into any of the modifiable. But hopefully, too, you maybe got a taste for the fact of that discrepancy, whether women are really more at risk. Are they more susceptible, or are they actually more resilient? Or is it both? Lastly, our work is supporting that there are specific genetic drivers, and these do play a role in how males and females develop and respond to disease pathology. And of course, this has huge implications. I believe in personalized interventions, personalized medicine, right drug, right time, and taking sex into account there. And with that, I will acknowledge our group, especially Dr. Holman here at Vanderbilt and the Vanderbilt Memory and Alzheimer's Center.
Awesome. Thank you so much. If anyone has any questions, now is the time. Feel free to put them in the chat. You can raise your hand. Thank you so much. This was a wonderful talk, so I'll let people ask their questions first. I also have a question actually to get us started. You kind of alluded to it towards the end of your talk, but I was wondering, with all these different sex-specific genetic findings, where do you see them translating into more clinically meaningful risk stratification and screening in the near future? Like, how do we then implement this specifically, to address these higher risk concerns for women?
Absolutely. So I think there's already a striking example right now with the current AD drugs that are out there. They haven't been well powered to look at sex different effects of the drugs, but they have noticed that it doesn't. I may have a slide, but I don't know. No, I don't think I have it in here. But the drug seems to have better performance in males than females. Confidence intervals are overlapping. Nevertheless, males seem to have react better to the drug than females. And this is especially important too when an ApoE plays a huge part too, right? So we know that there's like essentially a warning if you're ApoE4 positive that you can have reverse side effects. And so the interplay here, as we already know, the ApoE has stronger effects in females. How does that play into drug reactions and efficacy when we're looking at males versus females, and then ApoE and then genetic data on top of it? So I definitely think it's just ApoE right now. I think that's a major concern. Now, how it's going to translate for some of our other candidates, I think it's really interesting. We get a lot of pushback sometimes because of some of the variants we find have extremely small effects, right? As I mentioned, when we're studying GWAS, a lot of times those come out with very small effects. But there have been drugs that are put on the market. I think one example is there's some hypertensive and statin drugs and things that eventually came out. They found a single person that was resistant. They found out what gene was causing it. They exploit that and from a pharmaceutical level. And that small genetic effect size can turn into a very large therapeutic effect size. So that's the end goal.
Awesome. No, thank you so much. That answers my question. Thank you. Anyone else have any other questions?
Oh, come on, y'all.
Now's the time if you do. No, don't need to be shy. Sometimes it's hard with the Friday afternoons.
No, I get that. And you guys are on, yeah, post lunch, right? So, yeah.
I guess a specific question that I have. So a study that I believe Vanderbilt's working on, I know that most of the ADRCs are working on is the Clarity study,
Mhm.
kind of just across all the different centers. It's one of the studies that I work on. So I'm just kind of wondering, do you see the field kind of moving to more of this collaborative approach where all of the institutions tend to work on different studies together rather than it being kind of more individualized? And then how do you see kind of the benefit of being able to look at sex differences across all of the different ADRCs rather than just one?
Oh my gosh, such a cool question. Yes, I hope we're moving that way. I feel like that's the only way we're going to make major progress is by moving into groups like that. And Clarity is just a shining example of how to do that very well, right? And that was the impetus a lot for the PHC, for our phenotype harmonization consortium, right? There was lots of silos and lots of work being done, but because we weren't concentrated and put our efforts together, your sample sizes were small. And this is especially important for sex differences, right? Because half the time you're dividing your sample size in half when you're looking at males and females specifically, right? So yes, absolutely. It's a huge important aspect. Like we've got to get together and work together and have these larger efforts that cross ADRCs, that cross cohorts, if we're going to delve into these sex-specific effects, which I feel are important. They're small. They're not always there, but I feel like they're important and they actually can help us with some of the underlying biology. And as I already mentioned, have some therapeutic approaches too. So yeah, absolutely. Good plug for Clarity. They deserve it.
Thank you, Dr. Dumitrescu.
Awesome. Oh, I think Rick, you're unmuted.
Hey, Rick.
Hey Logan, great talk.
Thanks.
Do you think you have any sort of power to do a sex-specific PRS for resilience?
We have done a GWAS, a sex-specific GWAS for resilience. It came out a couple of years ago. We couldn't, the individual loci aspect of it, we were still underpowered, right?
Sure.
So a lot of our interesting actually findings came from when we looked at genome-wide covariance with other traits, right? So that's sort of like building a PRS in a way, right? So you're looking at all the genetic variants, and we looked at a couple of common traits, and one was heart. I forget what it was. It was a specific measure of heart function that had a very large sex GWAS, and we found that males had a higher correlation with. No, it was females that had the higher correlation. They were more resistant. The more resistant, genetically resistant females had a higher correlation with that heart trait. And these correlations, it's hard to figure out the direction of effect, but I think that's one thing that we can get out of it is general biology, what pathways are intersecting, which ones are not. One thing we're trying right now is using building PRSs or running these whole genome correlation analyses with sex hormones, right? So a lot of cohorts don't have that, or they're not measuring blood hormone levels, sex hormones. But they can be highly heritable, and there's huge GWASs out there of these sex hormones.
Did you,
Yeah.
in your new paper, did even with your silver model that you guys did, you probably have more power in that.
Right. So what Rick's talking about is our group is really interested, and I had so many things I wanted to talk about, but how we measure resilience, right? So this is the way, one way we could do it is how much your cognition, is it above or beyond what you would expect based on your your burden of amyloid and tau. You have a lot of tau, a lot of amyloid, your cognition should be lower, but if it's not, then you're more resilient. So we build these latent variable models of resilience. And, but again, a lot of them are based on use the gold standard with neuropathology. So that's, you're going to have your autopsy, which is, unfortunately, we're still, that's a lot of trouble. It's a lot of work to get the autopsy data. And so these biomarkers are a lot more convenient. So what if we can use kind of a silver standard model and use basic either demographics or some of these continuous traits, these continuous biomarkers instead. And it kind of works. I mean, it's signal through the noise, right? It's always what it seems like it comes down to in genetics is you get more, it gets noisy, but it's signal through the noise. Yeah.
I know sex was one of your variables in that, but wondered if you could do a sex stratified silver model and see if
Yeah, yeah, that's a great point to see if it actually makes a difference in the performance of even the silver model. Yeah. I mean, right. It was one of those demographic variables that is that we used in the silver models.
I'm sure it would. But I was just wondering from the GWAS standpoint, it might give you
Yeah.
might
No,
be able to get a sex specific PRS.
yeah. And we struggled with building the sex-specific resilience measure. We didn't know if we should build it in everybody and then stratify by sex, or do we build it within males and build it within females? Would that incur bias? So just that model building alone is kind of is a bit
Well,
tricky.
you got plenty to keep you busy for the.
Yeah. Yeah.
Awesome. If anyone has any last questions in our last couple of minutes, feel free to unmute and ask now. But this was a wonderful talk, and I had some emails that they couldn't make it, so I'm definitely going to upload that to the website, and that will be great for everyone to be able to look at afterwards.
Great. All right. Thanks for listening, guys. Have a good Friday. Have a good weekend.
Thank you so much. Appreciate it.